Okay, so our bodies do not have, or they have a finite amount of DNA, so we need to have a way to replicate our DNA. And so DNA carries our genetic information, as you guys probably already know. It needs to be copied every single time a new cell is made. So when you originally started out in your mom's tummy, you only were a couple cells, right? And you had to replicate, or really technically 2 cells, right? You had to replicate the DNA from those 2 cells in order to fully grow your body into all of the cells that it is now. So every time a new cell is made, you need to replicate the DNA. So DNA replication starts with helicase, I think it's actually how you pronounce it, helicase, which unwinds a part of a double helix. So it breaks apart the hydrogen bonds. All it does is adds energy here to just pull these apart. So now you have new parent strands, new single strands that are used as templates for new DNA molecules. Complementary base pairs come together. So more base pairs are going to be added in order to link these together. And a enzyme called DNA polymerase catalyzes the phosphodiester bond. So you have individual nucleotides coming in, and then DNA polymerase catalyzes the reaction to make new DNA. So you have one strand breaking apart, matching up, and making new strands. So the new DNA is going to be one template strand and one new strand. And then again, that could break apart and form more and more DNA. So let's look at our practice problems. What are the functions of the enzyme helicase and DNA polymerase? So helicase is going to be split DNA apart. And then DNA polymerase is going to be to form the new chains. So form the phosphodiester bonds. For the new DNA strands. Okay, and then how many hydrogen bonds link the following base pairs? So this is to reiterate everything that we've talked about, or what we talked about in the last video. How many hydrogen bonds link the following base pairs? A to T has 2 hydrogen bonds, G to C has 3. That's why it's stronger.